Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute. From today, we are going to learn the unit 2 of pharmaceutical biotechnology, where we are going to learn a lot of important things about genetic engineering, how you can do molecular cloning, you can use vector and all the aspects of our genome editing and other things. So, for today I am going to tell you about the cloning vectors, restriction enzyme and DNA ligase. What are these things? I am going to tell you in detail about molecular cloning, cloning vectors, why we need the cloning vectors, then what are the vectors are actually used in gene cloning, then what are the features of cloning vector, types, then what determines the choice of the cloning vector, and then restriction endonuclease and DNA ligase. These are the things we are going to learn on today. So, first of all, the biggest question is what is molecular cloning? Sir, at the beginning you have told us that you are going to teach us molecular cloning. So, my dear students, I hope all of you have heard lots of stories in our biotechnology, where scientists have successfully done cloning of several organisms or several small organisms or bigger organisms. So, you are aware about the cloning. If we have particular certain substance, certain features of a DNA or something, if we clone that thing, if we make it something similar, we call it as a cloning. Molecular cloning is the molecular level. So, usually in molecular cloning, we actually we need some vector, then we need some DNA fragment. DNA fragment is our interest, gene of interest we can say. If a DNA it having some quality, all of you are aware this DNA to mRNA will be created, and then from that mRNA ultimately protein will be developed. All of you are aware about these things, transcription and translation. Now, if you have some DNA, a good DNA, you want to express that DNA, so that you can get some good protein. So, in that case you will be taking the DNA fragment and you will be doing the construction of the chimera. You will be adding the DNA fragment in that vector. See how it is happening. Then you are actually putting that entire fragment or entire your chimera into a host cell. For example, we are actually using bacteria, E. coli or other kinds of bacteria. Then what will happen? Then multiplication will be happen. After that, when this cell it will divide, then multiple cells will be created. When multiple cells will be created, at that time whatever the DNA fragment was there, it will also express, and due to that you will be getting such kinds of protein. So, this is actually molecular cloning. Now, all of you have seen here in this area we have talked about vector and we have talked about the DNA fragment. That is the thing is our today's area of interest. So, what is vector? I have told you na vector. So, vector is a DNA molecule that is used to carry foreign DNA into the host cell, foreign DNA, this one. Okay. Then it has the ability to self replicate and integrate into the host cell. Also, these vectors have helped in analyzing the molecular structure of the DNA. Also, vectors can be a plasmid from the bacterium and a cell from higher organism or a DNA of a virus. See, let me erase. Yeah, see, plasmid from bacterium. We have seen in the previous case, we have actually taken the help of the bacteria, and also a cell from the higher organism or that is a DNA or a virus. I will tell you more how we can use various kinds of this thing called vector. What are the types of vector? I am going to tell you in my upcoming slides. You may also learn that the target DNA is inserted into the specific site of the vector and ligated by the DNA ligase. You have seen targeted DNA inserted into the specific site. This is the targeted DNA inserted into the specific site. See, okay. It was inserted into the specific site. We can tell more. If I tell you more, the vector is transferred into the host cell for replication. You have seen in the previous, and also the molecular analysis of the DNA has been made possible by cloning of the DNA, and the two molecules that are required for the cloning are the DNA to be cloned and the vector. Whatever we have seen in our previous slides, I am actually talking about these things, this and this. These two things are required. Then a cloning vector is a small piece of DNA taken from a virus, a plasmid or a cell, or a higher organism that can be suitably maintained into a organism and into which foreign DNA fragment can be inserted for cloning purposes. Okay. And most vectors are actually genetically engineered. See, if you take the help of some vector, you will have some goal. You will have some goal. For example, you try to make some vaccine. How your vaccine will be? What will be the structure? What will be the features of that vaccine? So, ultimately to get your desired product, you will design or you will take or you will genetically modify your vector according to need. Okay. Then if I tell you more, the cloning vector is chosen according to the size and types of the DNA whatever will be cloned. You can see here are various examples of cloning vectors I am giving. Okay. I am going to tell you more about these things. Okay. Every time you will be see these kinds of round shape pictures, these are nothing but some vector. In one part of the vector, for example, this side, one part of the vector, we are actually going to insert our particular that gene of interest, our DNA fragment. The vector therefore contains features that allow for the convenient insertion or removal of the DNA fragment in or out of the vector. For example, by treating the vector, the foreign DNA with restriction enzyme and then ligated with the fragmented that is after. After the DNA fragment has been cloned into a that is a cloning vector, it may be further subcloned into another vector designed for more specific use. See, nowadays we are actually using this kinds of thing. You are as a beginner, you should not be so much worried about this point. Okay, yes, scientists are doing because we need we are actually doing this genetic engineering because of our need. We have some need, that is why we are doing such kind of things. So, you can make entire clone. You can make entire clone, then you can insert it again another one. Now, the biggest question, why we do or why cloning vector? So, cloning vector is actually used as a vehicle to artificially carry foreign genetic material into another where it can be replicated or expressed. Okay, it is was like a vehicle. It is used to amplify a single molecule of DNA into many copies, you have seen in the previous. Then cloning vectors are DNA molecules that are used to transport the clone sequence into the biological host and tube, and without cloning vector molecular gene cloning is totally impossible. Now, vectors in cloning vector, what is the story, sir? Who is the scientist who discovered these things, and how today we got to know that yes, such kinds of things are available and we can use this cloning or vector. Scientists like Howard Ware, Kochi and other Arthur Rings, they are working in a Weyer's lab, University of California. They have recognized general cloning vector with unique restriction site. I will tell you more what is restriction site for cloning in foreign DNA and expression of antibiotic resistance gene for selection of the transformed bacteria, antibiotic resistance gene. Can you imagine? Today's scenario, it is a big problem. Senior professors like Professor Dr. Abhijit Majumdar, the director of NIT Pharmacy Institute, he has a great expertise, or he always aware us or inspire us to work on antimicrobial resistance. This is a big problem in present scenario, but you can imagine whatever the today's problem, whatever sir Dr. Majumdar sir and other academicians, scientists are encourage us to do research on that means called antibiotic resistance. These things are actually happening much more earlier. Okay, and that story, that resistance story is somehow related with our this thing called that is gene cloning. Okay. So, what happened in 1997, they described the first vector designed for cloning purposes. This is the name PBR322, a plasmid. It was basically a plasmid vector. Okay. So, you can imagine in last 30 to 40 years how much development it was happened. Okay. Now, if I tell you more, the vector was very small, it was 4 kilobyte in size and had 2 antibiotic resistance gene for the selection. So, today's problem whatever we are facing, today's that is a biggest challenge, that is this thing called antibiotic resistance. This story actually helped us to develop a new technology in 1977, can you imagine. Now, let me tell you more, what are the features of our cloning vector. First, origin of replication, sometimes we call it as ORI. This makes autonomous replication in the vector, and ORI, that is origin of replication, it is a specific sequence of molecule from where replication actually started. Okay. So, in every cloning vector, you will be getting some ORI. When foreign DNA is actually linked to the sequence along with vector replication, for that is a foreign desirable DNA also start replicating in the host cell. Another feature, there will be something called cloning site. What is cloning site? It is the place where the vector DNA can be digested and desired DNA can be inserted. Okay. See, for example, this multiple cloning site. Okay. So, it was the ORI in my previous slide I was mentioning na, ORI, this is the ORI, okay, this area, and this area is the multiple cloning site. So, it is a point of entry or analysis for a genetic engineering work. Basically, there are lots of enzymes will be worked together, it will be analyzed how it is happening, everything will be start from here. Recently, recombinant plasmid contain multiple cloning sites, okay, MCS, which having up to that is a 20 restriction sites. Now, another thing is called suitable marker. Suitable marker is a gene that confer resistance to a particular antibiotic or selective agents that would normally kill the host or prevent the growth. If I tell more, a cloning vector contain a suitable marker. See, marker is important, which confer on the host cell an ability to survive and proliferate in a selective growth medium containing the particular antibiotic. Okay, see, marker. Then reporter gene or marker gene, you can sometimes tell. So, reporter genes are actually used in cloning vector to facilitate the screening of successful clones by using the features of this gene that allow successful clone easily to be identified. Some features are present in cloning vectors used to be the blue or white section. These are the picture you can see. Now, if I tell few other features, first of all, it should be very short and small, then it should be compatible, it should be incompatible with the other vectors, because there are lots of bacteria and virus, other things will be there. If it may become incompatible, then it will be a very difficult. It should become high in copy number, then also it should able to express itself utilizing the host machinery, okay. And it should be also able to move under two systems, prokaryote and eukaryote. See, many of our cells are actually prokaryote, many of our new cells are actually eukaryote. So, it should be able to move in two systems. Okay. Now, what are the types of vector? Sir, at the beginning you told us, you promised us that you will be telling us what are the types of various vectors. Here is my slide, here is my answer. What are the types? First type is the plasmid. You already have seen from the beginning till now, almost 12 minutes we have spent. I was giving you all the examples of the plasmid based vector. Plasmid is the first. Second, bacteriophage. Many of you might be thinking, sir, bacteriophage is a virus, can you use it? Answer is yes. Then human artificial chromosome. Then this thing called phagamides. Then bacterial artificial chromosome. See, both the things are different, human artificial chromosome, bacterial artificial chromosome. Then cosmid, then yeast artificial chromosome. Why? Yeast artificial chromosome. Okay. Then retroviral vector, lentivirus, etc. we are actually giving. Now, first of all, what is plasmid and how you can use plasmid as a vector? So, plasmid is an autonomously replicating circular double stranded extra chromosomal DNA, which is physically separated from a chromosomal DNA and can replicate independently. See, let me tell you one very interesting story what is happening in recent times. I hope many of you have heard about the mRNA vaccine for SARS-CoV-2 coronavirus. Do you know about a great scientist called Kate Broderick? She was a great scientist in our present time, because she is the person, she is the scientist who was working in SARS-CoV-2 coronavirus vaccine. Okay. When they have got the genome sequence of SARS-CoV-2 from China, immediately they have started work, and the videos are actually available on BBC's website. Her interview is also available. They have shown us that how she has developed within 15 minutes. Within 15 minutes, she has designed a plasmid based vector and gene of interest to make some mRNA based vaccine. Okay. So, that is why these things are becoming so popular nowadays. If I tell you more, they are most commonly found in the bacteria, sometimes they are also present in the archaea or eukaryotic organism. See, plasmid all over everywhere. And if I tell you more, what will the size of plasmid? It may vary from 1 to 200 kilobyte. Okay. This is a typical scenario, typical workflow, how the things actually happening. So, for example, gene of interest. For example, I have one DNA, I have one DNA and I know its feature. I know these things will create one good vaccine. So, this is my gene of interest. Okay. I will take one bacteria, the bacteria plasmid, for example, bacteria F plasmid. I will take it and I will cut it. Okay. I will cut it by using the restriction enzyme. What is restriction enzyme? I am going to tell you in my upcoming slides. Then F plasmid and gene of interest will be inserted together, okay, like this. Now, this will be called as a BSC, bacterial artificial chromosome. It will be electrophoreted into E. coli cell. This is E. coli, this is E. coli. Okay. So, these things actually inserted into bacterial E. coli cell. Now, when these things will be expressed, it will express that thing, that vaccine or that substance, whatever the we are actually needing. So, that is why plasmid is very important. Okay. I hope it is very much clear to you all. If I tell you more, most of the plasmids are may clone used to be cloned DNA up to 10 kilobyte in size. Then many plasmids have high copy number and high copy number is useful as it is produce the greater yield of recombinant plasmid and sequence manipulation. Then however, low copy number plasmids may be also preferably used in certain circumstances, and yes, for example, when protein from cloned gene is actually toxic to the cell, in that scenario we may use the low copy number plasmid. Okay. So, this is something about the plasmid. Now, come to another type of vector called bacteriophage. I hope all of you might have seen this picture of bacteriophage, sometime you call it as a T2, T2. So, this is a very popular bacteriophage. It has created a great story, great success in the field of our biotechnology and pharmaceutical biotechnology. You know why? CRISPR, I am going to tell you more about the CRISPR in my upcoming lecture. The basic of CRISPR is actually start from here. CRISPR is a technology by using which you can edit your genome. Okay. Some scientist in China in 2 years back, he has that is produced one baby, gene edited baby by using CRISPR, though it was highly criticized from all over the world, and the scientist later on he was actually arrested. Okay. So, there are lots of stories with this particular bacteriophage, but for our interest let me tell you how you can use this bacteriophage as a vector. They are more efficient than plasmid for cloning of large DNA inserts. Then lambda phase or M13 phase are actually commonly used in bacteriophageal gene cloning. Then 53 kilobyte DNA can be packed. Can you imagine 53? Earlier it was only about 10 kilobyte. Then this the screening of phase that is a phase plex is much easier than screening of recombinant bacterial cloning. Another thing, this is something about the our bacteriophage. Another way you can do cloning, another vector you can use is called human artificial chromosome. You can see this is a human chromosome centromere. So, if you artificially manipulate a certain chromosome, if you artificially manipulate a human chromosome, you can also use that thing as a cloning vector. This is a common commonly used our the chromosome, but you can see this area is actually chopped off or it was actually reduced. Okay. So, you can see the telomere side actually reduced, that is replication side actually reduced. So, if you reduce its size or if you make it according to your need, then you can call it as a human artificial chromosome. Now, human artificial chromosome or HSC based vector often these vectors offer a promising system for delivery and expression of full length human genome or tiny size. It is a tool for expression studies or determining human chromosome function, and yes, these kinds of artificial chromosome avoid the limited cloning capacity and lack of copy number controlled and insertion that is mitogenesis caused by integration of host chromosome that plague viral vector. This is something about the HSC. Now, bacterial artificial chromosome. So, these are actually similar with E. coli plasmid vector. It is obtained from naturally occurring F plasmid, and these are used for the study of the genetic disorder, and this can be accommodated with a large DNA sequence without any risk. Okay. This is the bacterial plasmid, and if you this is actually E. coli. Okay, this is the gene of interest. Almost it is similar with the E. coli plasmid vector, almost it is similar, but in that case its size was very small, here size is slightly bigger. Another thing most popular, it is called yeast bacterial chromosome. Earlier we have used bacteria, here we are actually using yeast. Now, what is yeast bacterial artificial chromosome? So, yeast artificial chromosome is a human engineered DNA molecule used to clone DNA sequence into yeast cell. Okay, this is yeast cell. So, here we are actually making these kinds of things. So, YSC are often used in the connection with the mapping and sequence of the genome, and also this YSC cloning systems enable the cloning of DNA stretches from 50 to 2000 kilobyte. Can you imagine 50 to 2000 kilobyte? Earlier in our previous vectors we have seen up to only 10 kilobyte or 100 kilobyte. Here you are actually getting 2000 kilobyte. Segments of an organism's DNA up to 1 million have base pair in length can be inserted. This is a picture of YSC. This is yeast cell basically. This is a nucleus and this is the yeast artificial chromosome. Now, what determined the choice of vector? How you will determine during your work, how you will determine what that is a vector will be chose. So, first is the insertion size, then is the vector size, then restriction size and also cloning efficiency. Here I have given a chart that the size of the that is a vector. Okay. Let me again tell you, size of the vector. Now, I have told you several times that during our work we are actually using restriction and a nuclease. When plasmids are actually added with the gene of interest, we are actually using the restriction endonuclease. Sir, what is restriction endonuclease? This is nothing but it is a restriction and it is a enzyme that clips DNA into fragment at near specific recognition site within a molecule known as a restriction site. See, this is like a scissor, it will be cut a certain site of a DNA. These enzymes are one class of the border endonuclease group of enzymes. See, this is the picture, you can see restriction enzyme, it is also known as the molecular scissor because it will cut the DNA. See, this is the area where it is actually cutting because this is a targeted site. Here is a sticky end or this is our gene of interest. Okay. This is our gene of interest and both the things are joined together and you can see ultimately we are getting the recombinant DNA. Okay. So, the restriction enzymes are commonly used classified into 5 different types which differ in their structure and other things. If I tell you more to cut the DNA, we are actually using, and these enzymes are actually found in the bacteria or archaea or other things. Together the two processes are actually called as the restriction modification system, and more than 3600 restriction endonucleases are actually known, which represent over the 250 different that is subspecified. Over 3000 of this have been studied in detail, more than 800 of this actually available in the commercially. We are actually using huge of these kinds of enzymes. These enzymes are actually routinely used in DNA modification of laboratory, and also it is a vital tool in molecular cloning. You have seen without this kinds of restriction endonuclease, this molecular cloning it is impossible. You cannot cut a DNA. If you cannot cut a DNA, you will be never able to insert a certain DNA into a plasmid or other things. So, that is why it is very much important. Now, if I tell you more, inside the prokaryote, the restriction enzymes selectively cut up the foreign DNA in a process called restriction digestion. Meanwhile, host DNA protected by the modification of the enzyme, and together we call it as a restriction system, and that thing I have already mentioned. Here is a picture, already you have you are much more clear that how the things actually happening. And now let me tell you about the DNA ligase. I have a very short period of time, I am going so faster, so I am sorry for that. Now, DNA ligase, it is a specific type of enzyme or ligase that facilitates joining of DNA strand. See, by using restriction enzyme, restriction endonuclease, you have cutted the DNA, you have inserted DNA. Now, for insertion you need something some ligase enzyme which will make you or which will that is join the DNA strands together by using the phosphodiesterase bond. See, this is the picture, DNA ligase actually joining, both the things are actually cutted, it was a separate, this was separate, DNA ligase actually joining. It plays important role in repairing single strand break in duplex DNA in the living system. Some but some form of the DNA or ligase or maybe specify in repair the double strand break. Okay. You can see there is a picture, single strand break actually repaired by the DNA ligase using complementary strand or double helix. Also you can see the DNA ligase is used both in DNA repair or DNA replication. In addition, DNA ligase has extensive use in the molecular biology laboratory, where for recombinant DNA experiments. See, I will tell you more in the research application in my in my next few lectures, where I have in detail I have mentioned how you can use in the applied field. So, purified DNA ligase actually used in cloning to join the DNA molecules and recombinant DNA. So, today we have learned the molecular cloning, you have seen the cloning vector, why it is important vector in the gene cloning, what are the features, types, how we will determine or how you will choose a vector, what is resident endonuclease and what is the DNA ligase. All the things are actually associated with our entire learning of the genetic engineering. So, that is it for today. For next day tomorrow I will tell you that is recombinant DNA technology discovery, then what are the goals, procedure and few techniques are actually used in the rDNA. So, thank you all of you, thank you for joining. We shall meet in our next lecture. Keep learning, keep shining. All the best. God bless you all. Thank you.